Hello, my name is Gavin Sandall, and I conducted research this summer under the guidance of Dr. Margaret Saha. My project was a part of the ongoing research in the lab on bacteriophages. The title of my project is Characterizing and Discerning the Function of Antirepressor Proteins in Mycobacteriophages. Bacteriophages, or just phages, are viruses that infect bacteria. They're highly abundant, outnumbering bacteria tenfold, and they play a critical role in shaping microbial ecosystems. Phages influence carbon cycling and shape human health through the microbiome, while also holding high potential for gene therapy and bioengineering. Phages can undergo two distinct life cycles, the lytic cycle and the lysogenic cycle. In the lytic cycle, the phage hijacks the host machinery, replicates, and then bursts the cell open to release progeny. This allows for rapid amplification of the virus, but comes at the cost of killing the host cell. In the lysogenic cycle, the phage DNA integrates into the host genome and becomes what is called a prophage. The host continues to live and divide, copying the viral DNA each time. These so-called lysogens are often immune to reinfection by the same phage. Under stress, however, prophages can switch back into the lytic cycle. The switch between lysis and lysogeny is tightly regulated. The repressor protein maintains lysogeny by blocking the expression of lytic genes. Antirepressor proteins, on the other hand, can inactivate repressors and tip the balance towards lysis. A well-studied example is lambdaphage in E. coli, but less is known about these systems in mycobacteriophages, such as Doc McStuffins. With that background, the guiding question of my project is, how does the Doc McStuffin phage's antirepressor influence lysogeny in Mycobacterium smegmatis? More specifically, can we reliably establish and confirm an M-SMEG-Doc lysogen? Can we introduce plasmids carrying antirepressor constructs into the system? And finally, can we observe whether the antirepressor destabilizes lysogeny and promotes the lytic cycle? The first challenge was establishing a stable Mycobacterium smegmatis Doc McStuffin's phage lysogen. Through PCR and growth assays, I determined that many of these old cultures were either contaminated or no longer viable. To reestablish a new Doc lysogen in Mycobacterium smegmatis, I used the C phage's lysogeny protocol. I went through 2 rounds of purification, which included streaking, patch tests, and spot assays. I confirmed lysogeny using both culture PCRs and an immunity assay. Once confirmed, I scaled up the culture and prepared Doc lysates with titers close to 10 to the 9 plaque-forming units, which is a measure of the infectivity of the virus. This provided the lab with strong working stocks for future use. Next, I prepared the molecular tools needed for future experiments. I verified the presence of the Doc antirepressor plasmid construct through restriction digests, and I began testing the CPLA-mediated transformations to introduce the plasmid into the lysogen. I made significant progress in optimizing the transformation protocols for mycobacteria in general. Finally, I focused on working with DNA. I extracted genomic DNA from the lysogen using a phenylchloroform isoamyl alcohol protocol, obtaining good concentrations with solid purity scores. To further confirm integration, I designed primers which targeted the integration site in Mycobacterium smegmatis, which will allow us to check for proper lysogeny through polymerase chain reaction, or PCR. My methodology this summer involved rebuilding a confirmed lysogen system and preparing the plasmid and DNA tools necessary to test the Doc McStuffin's antirepressor going forward. Although I have not been able to test the Doc antirepressor directly, I accomplished several important milestones. I reestablished and confirmed through sequencing a clean lysogen system, prepared high-titer phage lysates, verified critical plasmids, and set up protocols for efficient future transformations. These steps effectively set up the project for future experimentation. This image shows an agarose gel confirming that the DNA that was extracted from the M-SMEG-Doc lysogen contains the Doc phage DNA and the M-SMEG DNA. The next steps will be to optimize transformations of the antirepressor constructs and test the effect of the antirepressor expression on lysogen stability. However, this summer research project can inspire many different future projects involving the characterization of phage proteins. Beyond this project, studying the Doc antirepressor has broader implications. It contributes to our understanding of how phages make developmental decisions, which is a fundamental question in microbiology. It also has roles in phage therapy and synthetic biology, as phage repressors and antirepressors are natural genetic switches that can inspire new circuit designs. Thank you for listening, and I'd like to acknowledge my advisor, Dr. Saha, my lab colleagues, and the Charles Center for funding my research.